Hello, my name is Ryoko Oi. I'm a researcher at G&L Kyosei Institute. This time, I introduce GADV protein world hypothesis by Dr. Kenji Ikehara. I am in charge of this narration. As well known, all organisms inhabit on the Earth under the fundamental life system composed of gene, genetic code, and protein. Legitimacy of the hypothesis on the origin of life should be judged by whether the formation processes of the three components, gene, genetic code, and protein, can be reasonably explained by the hypothesis or not. So the most important point would be to make clear how the life system was created on the primitive Earth. Out of three, formation of protein or its catalytic activity would be the most essential for the first life to emerge. But the first protein must be produced by random joining of amino acid because the sequence cannot be designed previously. Nevertheless, functional protein must be produced by direct random joining of amino acid. It is quite difficult to understand how the random polymerization could be converted to the synthesis of polymer with ordered sequence during repeated random processes. Inversely saying, it would be essential to know what caused the conversion from random to systematic polymer synthesis for making clear the riddle on the origin of life. GADV hypothesis can rationally explain the evolutionary process from accumulation of simple organic compounds on the primitive Earth to the emergence of life. Then how was the first functional protein created in the absence of any genetic function? Protein's order structure, pre-primary structure, made it possible to produce water-soluble globular protein before gene formation. Protein's order structure, pre-primary structure, is a specific amino acid composition in which even random joining of amino acid can produce water-soluble globular protein with a slightly more flexible structure than extant or mature protein at a high probability. Protein's order structure, pre-primary structure, holds the key for solving the riddle on the origin of life. That is, protein's order structure, pre-primary structure, or a specific amino acid composition in which random joining of amino acid produces water-soluble globular protein made it possible to convert from random process to synthesis of protein with ordered sequence. A pool containing roughly equal amounts of GADV amino acid, glycine, alanine, aspartic acid, and valine, or the specific amino acid composition, is one of the protein's order structures. So random joining of GADV amino acid could open the way to establishment of the first genetic system through the formation of water-soluble globular protein, or useful one, although only useless ones should be generally produced by random polymerization. Genetic code connects genetic function on DNA or RNA with protein synthesis, or actually triplet nucleotide sequence or codon with amino acid. In addition, The genetic code specifies a framework of amino acids which can be used in protein synthesis. Of course, even polypeptide chain, which was synthesized by indirect random joining of amino acids under the first genetic code, must be folded into water-soluble globular structure. This means that the first genetic code must encode amino acid in the simplest protein zeroth order structure. Ikehara et al. Have previously proposed GNC primitive genetic code hypothesis, suggesting that the universal or standard genetic code originated from GNC code encoding GADV amino acid. Random polymerization of amino acid would be carried out under the GNC code after the first genetic code was established. The polypeptide chain produced could be folded into water-soluble globular structure at a high probability because random joining of amino acids through GNC code is essentially the same as GADV protein synthesis by direct random joining of GADV amino acid. The first gene must be formed by random joining of nucleotide. However, genetic information never be produced by random joining of nucleotide. Because nucleotide sequence diversity is extraordinarily large. Of course, functional protein must be produced even under the first gene. Thus, the first gene must be formed by random joining of triplet GNC codons but not mononucleotide. The establishment of GNC genetic code made it possible to use only GADV amino acid for protein synthesis for the first time. The GADV amino acid sequence space 4 to the 100th equal 10 to the 60th encoded by the GNC code is much smaller than the full space 20 to the 100th equal 10 to the 130th of a protein composed of 20 amino acids. Therefore, foldable polypeptide sequences are contained in the GADV amino acid sequence space at a much higher ratio than the full space. The first life cannot live with only one gene encoding one protein. Thus, many genes must be effectively produced with the first gene. The first double-stranded GNC in parentheses sub-N gene could overcome the difficulty. After creation of the single-stranded GNC in parentheses sub-N gene, plural number of GADV proteins with the same amino acid sequence could be repeatedly synthesized by expression of the GNC in parentheses sub-N gene, although the GADV protein was only immature protein with random GADV amino acid sequence. In order to create the first double-stranded GNC in parentheses sub-N gene, many difficulties must be overcome, such as establishment of the first GNC genetic code, the first single-stranded GNC in parentheses sub-N gene formation by joining of GNC codon in oligonucleotides. The first double-stranded GNC in parentheses sub-N gene was created by complementary strand synthesis of the first single-stranded gene. The formation of the first double-stranded GNC in parentheses sub-N gene made it possible to evolve immature and rather flexible protein with a weak catalytic activity to mature rigid protein with a high catalytic activity for the first time. Successively, both homologous and entirely new genes could be produced from sense and unsense sequences of the first double-stranded GNC in parentheses sub-N gene after gene duplication, respectively. This makes it possible to produce homologous protein in a protein family and entirely new protein or the first family protein from sense and unsense sequences of the first double-stranded GNC in parentheses sub-N gene, respectively, after gene duplication. Life is a high-level concept associated to systems of molecules, cellularity, self-maintenance, reproduction, and so on. Probably all these features are linked to protocellular systems rather than few replicating molecules. However, many genes fortunately could be produced by using the first double-stranded GNC in parentheses sub-N gene, because both base sequences on sense strand of the gene and the corresponding unsense sequence are GNC codon sequences. Thus, both GNC in parentheses sub-N gene and its unsense GNC in parentheses sub-N sequence could code for GADV proteins with similar amino acid composition but quite different amino acid sequence each other. How did protein growth order structure play a role in establishment of the fundamental life system? Protein growth order structure, GADV amino acid, made the first genetic system composed of GADV protein, GNC genetic code, GNC in parentheses sub-N gene as follows. Water-soluble globular GADV protein as an aggregate of GADV peptides could be produced. Stage 1. By direct random joining of GADV amino acid. Stage 2. By random joining of GADV amino acid through primitive tRNAs. Stage 3. By using single-stranded GNC in parentheses sub-N gene, which was formed with random arrangement of GNC codons. Thus, Ikehara has proposed GADV hypothesis, assuming that the evolutionary stage to the emergence of life progressed to produce more complete GADV protein than before, step by step. I explained the evolutionary stages to the emergence of life in more detail. Water-soluble globular GADV protein, actually aggregate of GADV peptides, could be produced by random joining of GADV amino acid at a high probability, because GADV amino acid composition is one of protein growth order structures. The first GNC genetic code encoding GADV amino acid could be established because GADV amino acid composition is a protein growth order structure. The first single-stranded GNC in parentheses sub-N gene encoding GADV protein could be formed through joining of GNC codons. The first life emerged after a double-stranded GNC in parentheses sub-N gene, because various homologous and entirely new genes could be produced from sense and unsense sequences of a double-stranded G and C in parentheses sub and gene, respectively. I believe that life emerged from GADV protein world, because GADV hypothesis can explain reasonably the formation process of the genetic system composed of gene, genetic code, and protein. Of course, it should not be concluded that life emerged on the primitive Earth as the hypothesis assumes, even if the evolutionary process to the emergence of life can be explained by the idea rationally or without large contradictions. However, inversely, if the evolutionary process cannot be reasonably explained with an idea, it could be concluded that the idea is wrong. Thus, I would like to say that life did not emerge from RNA world.